Can you start by telling us your name, your institution, and your role?
Hi, yeah, I'm Manda Arbab. I'm an assistant professor at the Translational Neuroscience Center at Boston Children's Hospital and Harvard Medical School.
And then can you describe the SCGE research project you're working on in a couple sentences?
We are developing in vivo therapeutic base editing and prime editing strategies for several severe neurological conditions. And our Trailblazer project aims to translate a base editing strategy that we previously developed for spinal muscular atrophy for use in patients. And then other projects as part of our grant focus on treating trinucleotide repeat disorders, including Huntington's disease and Friedreich's ataxia, as well as Rett syndrome.
And then what specifically do you work on within the research project?
I lead the Trailblazer project, which is on SMA, spinal muscular atrophy. So I oversee the collaborative efforts between our lab, Jackson Labs, UT Southwestern, and the Broad Institute to essentially produce data that would support an IND study. And then in our own lab, we're also continuing with optimizations of this leading strategy for use in patients to improve things like safety and efficacy to whatever degree that is still possible at this point. And I'm also PI on two of our other SCGE projects where we're completing development of both a base editing and a prime editing approach for the trinucleotide repeat disorders, Huntington's disease and Friedreich's ataxia.
Great, thank you. And then what's your favorite thing about either this research or other research that you work on?
Probably that I think that we're really at a very exciting time in developing gene-based therapeutics. It's a very new area, and science and technology in our field has really advanced at a very high pace in the last 10 years. We're doing things now that we could have only really dreamed of doing 10 years ago. And the fact that we're also now we're at a phase where we're taking these things to the clinic at an accelerated pace is extremely exciting. So yeah, I'm thrilled to be a part of that.
And then what's something cool, interesting, or unexpected that you found in this research?
Well, I've been working on spinal muscular atrophy for a very long time, and it always been my sort of career goal to be able to achieve some sort of rescue of that in a mouse model to hopefully one day be a therapeutic. So it's very exciting to feel like we're at that phase right now. And also recent developments that we've had in our trinucleotide repeat projects have been extremely exciting, where we've been able to demonstrate for the first time how doing a specific form of genetic intervention for these diseases can actually halt this sort of key underlying cause of the disease, which is the repeat expansion. So that's been very exciting.
What's an advancement that you think will happen in genome editing in the next 10 years or so?
Well, something I certainly hope will happen, and there's a lot of exciting efforts, I think, toward that goal, is trying to insert and replace larger fragments of DNA, ideally even gene-sized fragments of DNA. If we could, in a site-directed manner in cells at high efficiency, insert or replace entire genes, I feel like that, above and beyond anything else, will really revolutionize the field, both in research and in medicine.
How do you engage with patients or patient advocacy groups within your research?
So I work at the Translational Neuroscience Center at Boston Children's Hospital, where there's a large focus on sort of interacting with patient organizations. So I interact with several different patient organizations. I have done some advising. I'm on an advisory board for some of these sorts of disorders. So it's mostly on that level that I interact with patient groups. And then, occasionally I'm contacted by patients who or parents of patients who have genetic diseases, and then we try to work together with the resources that we have at the Translational Neuroscience Center to see how it is that we can, if we can, and how we can best serve them.
Is there anything else that you would like to say about your project or your research or the SCGE consortium more broadly?
I think that this is a fantastic way of the NIH to bring all these different groups together. First of all, I think it's very well organized, and I'm very uh, thrilled with how our group internally is sort of able to progress our projects, and I think it's going very well. But more broadly for the consortium, I also think it's wonderful how all these different groups are being brought together, which is as far as networking and being able to think about core key outstanding issues in the field in general, and being able to translate this to the clinic. It's bringing a lot of good people together to think about these issues together, such as, how do we begin to interact with the FDA? Are there things that need to change from a regulatory perspective to be able to translate these new therapeutics to the clinic safely, properly, and more rapidly in the future? So yeah, I'm very happy to be contributing and to be a part of all that.